Thank you very much. I'm really, really glad to be part of this organization as such, and this is an excellent meeting so far. And I'm very glad to have come and really enjoyed the sessions and the discussions that we really miss in the bigger meetings that are very helpful for the basic scientist. Thank you. Sounds okay. All right. Okay, thanks once again. And I'm going to start here. Now we have targeting proteasomes. We have a couple of new, not new anymore. We have a couple of proteasome inhibitors which are FDA approved now, and we have others that are in clinical trial. All of these agents, they in fact, the target is proteolytic activities within the 20S core protein, beta 5, beta 1, and beta 2. Now, recent studies have really started exploring the alternative strategies to really target the protein degradation process at the level of proteasome itself or upstream of the proteasome at the level of ubiquitinating or deubiquitinating enzymes. And indeed, we have multiple sites of pharmacological intervention within the ubiquitin proteasome system shown here. We have E1, E2 enzymes, we have E3 ligases, as well as we have deubiquitinating enzymes called DUBs. And we are really interested in elucidating the role of DUBs in the biology of multiple myeloma. So what are DUBs? Our genome encodes about 100 putative DUBs, and they can be categorized in 5 distinct families shown here. And the USP, which is ubiquitin-specific proteases, are the largest family here. Their main function is to remove the ubiquitin from the substrates and allowing the stabilization of these substrates, as well as they maintain the ubiquitin homeostasis in the cellular context. DUBs have been linked to the pathogenesis of various diseases, including cancer. We have begun to analyze DUBs in our multiple myeloma models, and we have started doing DUB siRNA screening against myeloma cells in a high throughput fashion. We have identified so far a number of drugs, for example, USP14 and UCHL5 are two among those. These two DUBs are of particular interest because they regulate, they actually, in fact, reversibly associate with the 19S regulatory particle of the proteasomes. And in doing so, they remove the ubiquitin from the polypeptide chains, allowing for the polypeptide entry into the proteasome and its degradation. So our collaboration with Dr. Linder at the Karolinska Institute have allowed us to discover a small molecule inhibitor of USP14 and UCHL5 named BAP15 here. So if you block the USP14 and UCHL5 here, it leads to accumulation of large-sized polyubiquitin conjugates that can be reflected as a high molecular weight bands in this plot. So we did different assays to establish the specificity of BAP15 against the DUBs, specifically USP14 and UCHL5. And I'm going to show you examples, 2 or 3 assays that we have done here. This is a simple assay and purified protein enzymatic assays ex vivo done. We utilize recombinant GFP tag to the ubiquitin, which in the presence of active 19S regulatory proteasome cleaves GFP and ubiquitin that you can then measure in an immunoblot shown here. This is a control. This is a recombinant GFP ubiquitin band, and this is a vehicle-treated band where you cleave the GFP, but this band disappears in the presence of BAP15. That happens in a dose-dependent and a time-dependent manner, and that's also true for blockade in the ubiquitin tetramer chain shown at the bottom. So these data suggest that BAP15 indeed blocks the deubiquitination process here. Another assay that we use is called UBAMC, cleavage activity assays. These assays are also classical traditional assays to measure the DUB activity overall. And what is shown here in this graph on the left side is a % DUB activity. We incubated 19S protein again in the presence of different concentration of BAP15, you could see there is a significant inhibition in the activity, overall DUB activity, whereas BAP15 does not affect any other DUB here. Just not moving, could you help? Here. Okay, so here is the third assay that we did to establish that indeed the BAP15 targets USP14 and UCHL5. We used the HA-tagged ubiquitin vinyl sulfide probe, and this probe is a competitive probe that binds to the active cysteine residues within the DUB active domain. So what you see here is that BAP15 was able to decrease USP14 activity, evidenced by the decrease in the labeling of the UBVS probe, and so is true for the UCHL5. So what we've shown so far is that BAP15 can target 19S DUB overall activities, and it can block USP14 and UCHL4. So does this blockade in fact affect the overall cellular proteasome function? Does it block the protein degradation process? For this, we did this kind of assays where we used a reporter cell line expressing ubiquitin yellow fluorescent protein that is constitutively targeted for proteasome degradation. And if you take this cell line and treat it with AP15, there is a dose-dependent accumulation of our reporter protein, suggesting that proteasome function itself is blocked now. Interestingly, we do not see the blockade in the proteolytic activity of the proteasomes, as is shown here. All three proteolytic activities, chymotrypsin-like, trypsin-like, and caspase-like activities, when examined here, had no decrease in their activity, whereas bortezomib served as a positive control and showed inhibition in the activity. So this drug inhibits the DUBs. It inhibits the cellular proteasome function, but it does not affect the proteasome activity. We then next directly examine the effect of BAP15 in our multiple myeloma cell line models, patient cells, as well as in our animal models. So shown here is a simple MTT assays, and we have various cell lines, and as we observed that BAP15 consistently decreased the viability in all cases, including the cells which are bortezomib resistant too. And same holds true for the patients shown on the left. There was a significant decrease in the viability in all cases, and the cells that were obtained were from the patients who were progressing while on lenalidomide and bortezomib therapy. We do not see a significant effect on the normal cells here, suggesting that it's likely a good favorable therapeutic index. We then examine the mechanisms of how BAP15 is actually triggering apoptosis in myeloma cells. And shown here are a couple of plots showing caspase cascade activation, caspase 3, caspase 8, and caspase 9, their activity. And when you incubate the cells with the pan-caspase inhibitor, you actually rescue the cells from BAP-induced apoptosis. And these data suggest that it is a caspase-dependent mechanisms. We also found that BAP15 in fact induces G2M growth arrest, and as is evidenced by a decrease in number of molecules associated with the G2M growth arrest here. Interestingly, we also examined unfolded protein response here. As for bortezomib, it similarly induces induction of phospho-EIA alpha, CHOP induction, and phospho-IRE alpha. We then went on to examine effect of BAP15 in our in vivo model. And shown here are the tumor volume curves, bioluminescence as we determined from here or measured from here. BAP15 was prevented the progression of the tumor shown in red here in mice, which was also associated with prolonged survival. And tumor resection from these mice indeed showed accumulation of ubiquitin, which indicates that indeed this is a proteasome inhibition in vivo by the drug. Finally, we also examined various combinations of AP15 with available drugs with us. And shown here is an example. This is a classical isobologram analysis that we utilize to assess the combination degree of two agents. And we calculate the CI value, which is combination index, which is less than 1 indicate synergy. And in this case, we found that BAP15 synergized with lenalidomide efficiently. So the take-home message is very simple here, that we do have a drug, a novel agent, that by virtue of its ability to block the 19S dubs here, can block the overall proteasome function without affecting the proteolytic activity. Now, this has a significance. The reason is there are reports or controversial reports that resistance to the bortezomib could be due to some of the defects within the 20S core protein, whether it is 3 proteolytic activities or other proteins here. But this effect is override. There is an override here if you use this drug. So nonetheless, you are still able to block the overall proteasome function. So this is a new class of proteasome inhibitors that we are talking about here, and I hope that we would see a good clinical results. That is coming up. We have started a clinical trial later this year. Everything has been designed, so we're hoping good results from this new agent. I'd like to thank my mentor, Ken Ederson, and these are postdocs associated in my team. Their work primarily is Etienne and Madhvi, Deepika. They've done this work. And Nupur, Paul, and Nikhil, they have been providing clinical samples for our studies. And Stig Linder collaboration at the Karolinska Institute, very helpful. Thank you very much, and I will be happy to address any questions.